Question 5 2022. Which of the following statements regarding enzymes is correct? 1. Activators affect the function of enzymes by binding to active sites through covalent bonds. 2. Shape of the active sites of enzymes changes due to temperature higher than the optimum level. 3. Many competitive inhibitors bind to active sites of enzymes irreversibly and change their shape. 4. Toxins bind to enzymes reversibly through covalent bonds. 5. Coenzymes are proteinous components, which are permanently or temporarily bound to enzymes. An enzyme is a macromolecule, which acts as a biological catalyst. Enzymes are produced in living cells. Enzymes are globular proteins with tertiary structures, different structures of proteins, primary structure, secondary structure, alpha helix, beta pleated structure, tertiary structure, quaternary structure. Each enzyme has its own sequence of amino acids. Enzymes get their globular structure by different types of bonds present in their side chains, such as hydrogen bonds, disulfide bonds, ionic bonds, and hydrophobic interactions or van der Waals interactions. This complex 3D structure gives the enzyme many of its properties, factors that affect rate of enzymatic reactions, temperature, pH, substrate concentration, enzyme concentration, and inhibitors. Inhibitors can be classified as competitive or non-competitive inhibitors. Competitive inhibitors bind to the active site of the enzyme. Non-competitive inhibitors bind to a place other than the active site, and changes the shape of the active site of the enzyme, so it cannot bind to substrate. Allostric regulation of enzymes. In many cases, the molecule that naturally regulate enzyme activity in a cell behave like reversible, non-competitive inhibitors. It may result in either activation or inhibition. Regulatory molecules bind to specific regulatory sites, other than the active site, via non-covalent interactions and affect the shape and function of the enzyme. Allostric regulation of enzymes can be classified as activation inhibition, cooperativity, and feedback inhibition. Some enzymes are made of two or more subunits, and usually their allostric sites are located where the subunits of enzyme connects. Allostric activation of enzymes. Subunit of an enzyme, before activation, substrate molecule, activator. Shape of the functional active site of the enzyme is stabilized when an activator bound to the allostric site. Subunit with altered active site, due to activator. The other active sites are also stabilizers, due to this activation, since the communication among the subunits is rapid. Substrate molecules are easily bound to the active sites due to this activation, causing the reaction to occur. Accordingly, the first statement is wrong. Because activators are bound to allostric sites called regulatory sites of enzymes, not to active sites. Other types of allostric regulation of enzymes. Allostric inhibition. When an inhibitor bound to the regulatory site, the shape of the active sites of the subunits are changed, as the reaction cannot be taken place. Cooperativity. In cooperativity, binding of one substrate molecule stimulates the binding of other active sites. Feedback inhibition. The metabolic pathway is stopped when a molecule of end product binds to the enzyme and acted as an inhibitor. Active sites of enzymes. Active sites of enzymes are substrate-specific, because the shape of the active site of an enzyme is complementary for the shape of the substrate. If shape of the active site changed, the substrate cannot be fixed with the enzyme. Shape of the active sites of enzymes can be changed due to temperature or non-competitive inhibitors. Shape of the active sites of enzymes changes due to temperature higher than the optimum level. Influence of temperature on enzymes. The rate of enzymatic reactions increases gradually when the temperature increases. The optimum temperature range for enzymes is 35 Celsius to 40 Celsius. Rate of reaction increases up to 35 to 40 Celsius. When the temperature increases further, rate of reaction falls down rapidly. Because ionic bonds, hydrogen bonds, and weak chemical bonds are broken due to higher temperatures, and shape of the active sites are changed. This is called denaturation of the enzyme. Kinetic energy of molecules is increased when the temperature increases. This will enhance the colliding probability of both enzyme active sites and substrate molecules, increasing the rate of reaction too. Rate of reaction falls when the temperature increases over the optimum temperature, since the enzyme is denatured. The second statement is correct therefore. Competitive inhibitors are similar to the substrate molecules in shape and nature. They compete with the substrate molecules to bind with the enzyme. Available active sites for enzymes is reduced. Due to this competition, rate of reaction decreases therefore. This condition can be prevented by increasing the substrate concentration. Accordingly, many competitive inhibitors bind to active sites of enzymes irreversibly, but they do not change the shape of active sites. The third statement is wrong therefore. Toxic compounds are bound with enzymes irreversibly, not reversibly. The fourth statement is also wrong therefore. Enzyme. Coenzymes are non-portentous molecules which bind with enzyme temporarily or permanently. They are not proteinous molecules. Enzyme. Coenzyme. Substrate. Enzyme-substrate complex. Accordingly, coenzymes are non-portentous molecules which bind with enzyme temporarily or permanently. They are not proteinous molecules. The fifth statement is also wrong therefore. Accordingly, the correct statement about enzymes is the second statement. Because shape of the active sites of enzymes changes due to temperature higher than the optimum level. Answer 2. Thank you for watching. Subscribe for more such videos.